This is Tiffany Tulau, week 2. I'm going to be doing my bacterial enumeration and dilutions plate counts lab. And I will begin now. As you can see, I have some of my equipment on, gloves. I might be putting my mask on. And then I'm going to go over my materials. See, I've gathered all of my materials needed for this experiment. Those are my pre-poured agar plates. And I will be working now to start the experiment. As you can see, I have sterilized my pipette and used it to put 2.25 milliliters of distilled water into each of the labeled containers here. Here's my yeast. It's sitting and starting to bubble. And ferment. Alright, I'm cleaning my workspace with the 10% bleach mixture that was created to sterilize our workspaces. Here I am washing my hands with soap and warm water prior to the beginning of my experiment. I will now pipette from my sterilized pipette 0.25 milliliters of the Cerevisiae solution into the yeast tube that is labeled 10-1. As you can see, 10-1. .25 into the solution. This is to make sure the organism is completely out of the pipette. We'll now begin the dilution process further, taking 0.25 mLs from here, the first test tube, and transferring it into the test tube marked 10-2. Making certain that it is completely gone from the pipette. And I will continue this for each tube all the way up to 10-6. Now rinsing my pipette in distilled water to ensure all the organisms are completely gone from my pipette. And I will start the next portion of transferring 4 drops for 0.125 mLs of each solution from the test tubes into its corresponding dish. So for 10-1, I will take 4 drops see 1, 2, 3, 4. I will now cover and swirl the plate to spread the solution. Making sure that it is evenly covered. And I will do the same for the next 6 plates. Starting next with 10-2. Here I am coming to the completion of the last 2 test tubes and agar platings. 3, 4. Mixing my pipette. Cover that one and swirl. And lastly, 10-6. One, 2, 3, 4. Now I'm going to prep my plates for storage. I will store them for the first 12 hours with the agar plates right side up, and then after 12 hours I will switch them after the liquid has absorbed into the agar. I have now moved my plates into my incubation area and will be leaving them in there for 12 hours at first, flipping them over, and then another 2 days. For my cleanup for this portion of it, I'm using hot water and soap with my brush to dispose of the bacteria. All the rest of it can be disposed of in the sink. I will dry these appropriately and store them for my next project. Me too, Lau. It has been 48 hours. I am about to begin the second exercise of our aseptic technique in culturing microbes video. I have cleaned my surface, I have my gear on, and I will then show you how I have everything set up with the candle. My plate, I have my specimen, everything ready. I'm going to light my candle and begin the smearing portion of the exercise. I'm now taking my sterilized inoculation loop out, shaking it dry. Going to remove the lid from my broth with the loop still in my hand. I will then sterilize the lid and the flame of my culture. Very carefully to not touch the loop. I will then gather some specimen. I've submerged it inside the broth. And I'm now going to transfer the specimen onto the plate here in one quadrant. in the zigzag motion. These plates are smaller than I'm used to, so it's a little bit difficult. Now we're going to place the lid on the agar plate and put the loop back into the cup of alcohol. I'm going to reflame the top of the lid. And replace the cap. Throw it to the side. We'll now take the sterilized loop and turn my plate to do another quadrant. Inoculation, and then back again in the alcohol. Wait for 20 seconds. And I'm going to shake this again. Do the same in a different quadrant. Back in the alcohol, place the lid. Try it one more time. Shake again. And then doing the last quadrant. Back in the alcohol. I plan to repeat those steps for the next 2 specimens of microorganisms and then put them in to incubate and let's see how they grow. I cleaned my workspace, have all my gear on. This is, as you can see, dilution of yeast 1. Let's go somewhere in the middle. This is diluted to 4 times, you can kind of see. And then lastly, we have the dilution of 6. You can see that there is very minimal growth there. So this concludes the video portion. I will go on to do my calculations and complete my lab report.